This module is the continuation of the fed batch culture kinetics. So this module is the fourth module of fed batch culture kinetics. So as we discussed in previous module of the various strategies of fed batch, and then how we maintain the quasi-steady state during the fed batch. So when that mu is equal to D. So the D is directly effect on mu and different other parameters of the fermentation process. So as we already discussed, that PERT expressed the change in product concentration in variable volume, especially in fed batch culture. So the dP over dt is equal to qp dot x minus dp. Mean if there is a product, basically this equation is the continuous culture equation, but PERT also utilize this equation for the fed batch culture too. So we will discuss in detail in later on. So if we talk about the product concentration change according to the balance between the product rate and the dilution by the feed, so the dilution rate. So if we say that fed batch quasi-state change over the time of the fermentation, mean if mu is not equal to D, so then there is a drift in mu or a D with respect to D. So if we cannot balance between these two rates, production rate as well as the dilution rate, then the steady state can be disturbed. So the product concentration in fed batch system over the time of the fermentation depend on the relationship of Q, P and mu, which we called as specific growth rate. Hence we can say that it totally depend upon D, which we called as dilution rate. So the if QP is strictly growth related, mean our product is growth related, then it will change as mu with D. So if mu and D is disturbed or a change, then we can say that our product associated with the biomass will also change. So the product concentration in such continuous fashion when there is mu is equal to D and there is a constant increase in the product, as we say, when our product is directly associated with the biomass. In previous module we see that when there is a variable volume and mu is equal to D, then there is a constant concentration of the biomass. So we can say here the same way the product concentration also remain the same. So as much as the product concentration increase, what happen? Due to the dilution, volume also increase. As an example, we see if there is a fermentation vessel and here is a 5 liter volume and there are 5 organisms, and the 1 liter of the addition of the medium, and that become a 6 liter total volume, and during that the addition of the 1 liter medium into the fermentation vessel, there is the increase of 1 cell, and then the total number of cells is 6. So when we see that 5 organism in 5 liter, so 1 organism per liter. So when there is addition of 1 liter volume, so by the same time there is 1 more organism. So the 6 organism per 6 liter. So the ratio remain the same. By the same way, if the product concentration remain the same in fed batch quasi-state, so we can say that the QP is constant and independent of mu, specific growth rate. So then the product concentration will the start of the cycle when dP is greater than QPX. but will rise with time as D decreases. So as the D will decrease, mean dilution will decrease, then the product concentration will increase. So if the dilution will increase, then product concentration decrease. So it directly associated with the D. So we can easily say that if our product is directly associated with the biomass, so the same way when the dilution is equal to the mu, then X remain the constant and the P will also remain the constant. If the D will increase, then the biomass concentration also decrease and the product concentration also decrease. But if the D will decrease, mean dilution rate will decrease, then what happen? X will also increase and P will also increase. So if the QP is related to the mu in a complex manner, then the product concentration will vary according to the relationship, as I already told you. Thus, the feed strategy of the system would be optimized according to the relationship between QP and the mu. So overall, to run the fed batch culture in a variable volume, our main focus on the addition of the limiting substrate by knowing at what time what substrate will be depleted. Keep in mind a very less change in the volume. So mostly when we say as an example, when we have a culture of a baker yeast fermentation, in the start we having a small amount of the medium and we have the inoculum. And there is a continuous feeding of the medium and the yeast grow. With respect to the mu, we are adding and there is a gradual change into the medium. So as well as the fermentation volume will increase in the fermenter, what happen? The biomass will also increase. So that we can run the fed batch until to that limit up till the fermentation vessel allow us. So as I already told in previous module that we can only use the 75% of the total fermentation vessel volume. So the remaining is needed for head space, which is very critical during the fermentation process.